Welcome to your CAPT Bootcamp presentation for mitosis and meiosis. In this presentation, you will learn the reasons for cell division in your body, the stages of mitosis, and the reasons for meiosis and the stages of meiosis. Mitosis is the process of making new body cells. So making new cells in places like your skin, your bones, your digestive system, any type of body cell. And the reasons for mitosis are repairing damage done to the human body through wounds or injury, facilitating the exchange of materials or making nutrients and waste easier to exchange, and the growth of the organism overall into adult form. The cell cycle is divided into three major parts: interphase, mitosis, and cytokinesis. Interphase is where the cell grows and prepares itself to divide and makes a copy of its DNA so that the two new cells are identical. Mitosis is where the cell actually divides in half, and cytokinesis is where the cytoplasm and organelles are divided. Interphase is where the cell is preparing itself to divide. So it's divided into three steps: G1, S, and G2. The important step that you need to know is here, S phase, where the DNA actually makes a copy of itself and forms what are called sister chromatids. The entire point of mitosis is to divide the two sister chromatids that are made during S phase into two new cells. Here's a picture of what DNA would look like under a microscope. Before cell division can happen, the DNA is going to coil up and form what are called chromosomes that are visible under the microscope. This picture is showing you two sister chromatids that are identical copies of DNA that were made in S phase. They're held together by something called a centromere. That'll become important later. Again, each chromosome makes an identical copy of itself called a sister chromatid. Mitosis is the actual division of the nucleus in order to make two new cells that are genetically identical. The first phase of mitosis is something called prophase. During prophase, the chromosomes coil up and become visible. You have two structures, the centrioles, that are going to move to opposite poles or sides of the cell, and those centrioles are going to create something called spindle fibers. You're also going to have the breakdown of the nuclear envelope. Here's a picture of what a cell in prophase would look like. These structures here are the centrioles. These are the spindle fibers that are made by the centrioles. Here's the nuclear envelope, and each one of these is a chromosome with its identical sister chromatid attached to it. The next stage of mitosis is something called metaphase. During metaphase, the chromosomes are going to line up at the center of the cell at a place called the metaphase plate. It's sort of an imaginary line. Then one spindle fiber from each pole is going to attach to the centromeres, and the centromeres, or the spindle fibers, are eventually going to pull the sister chromatids apart. This is going to be important because this is how each new cell is going to end up with an identical copy of the DNA. This is a cell in metaphase, and here you can see the centrioles again, the spindle fibers, and then one spindle fiber from each pole is attached to each set of sister chromatids. So you can see there's one from each pole attached to each set of sister chromatids. And those sister chromatids, here and here, are going to get pulled apart in opposite directions. During anaphase, the spindle fibers that are attached to the centromeres are going to start to pull the sister chromatids apart, and the chromosomes are going to move to the poles or the opposite ends of the new cells. So here's a cell in anaphase, and you can see each sister chromatid going to an opposite pole of the cell. Eventually, those identical copies of the chromosomes are going to reach the poles of the cell, and the cell's going to go through what's called telophase, where the chromosomes are going to uncoil and reform chromatin. You're going to get a new nuclear envelope, and the spindle fibers are going to break down. So in other words, you're ending up getting two new cells. Here's a picture of what a cell in telophase would look like, and you could see that each new cell is going to end up with identical DNA. During cytokinesis, something called a cleavage furrow is going to form and start to pinch the cell in half. And this is where the cytoplasm and new organelles are going to get divided. Each new cell is called a daughter cell. And this is how this happens in animal cells. In plant cells, they're going to form something called a cell plate in the center of the cell first. So here in an animal cell, you can see the cell membrane pinching in half and forming this cleavage furrow, which will eventually go all the way across and form two new cells, complete with identical sets of DNA. Plant cells have to form this cell plate in the center in order to make new cell wall on each side. Meiosis also has to happen in organisms that are sexually reproducing organisms. So sexual reproduction requires genetic material from two different parents in order to create one new organism. So meiosis is the process that's creating reproductive cells such as egg and sperm. It's going to reduce the number of chromosomes in a sex cell to half of the number that are in the regular body cells so that sexual reproduction can happen. So your body cells or somatic cells are what we call diploid, meaning they have two complete sets of chromosomes. So each somatic cell has homologous chromosomes, matching pairs where one chromosome came from the mother and one from the father. Sex cells, on the other hand, are haploid, meaning they have only one set of chromosomes. This is how sexual reproduction happens. So a haploid sperm meets a haploid egg, and you get a diploid ewe, or a diploid organism. This is a chart showing you the haploid and diploid numbers of different organisms, including humans. So humans, all of your body cells have 46 chromosomes, and your gametes have 23. Some other common numbers that you would see in other organisms like the garden pea, the fruit fly, a tomato, a dog, a chimpanzee, and how they compare to the human. Since meiosis is reducing the number of chromosomes to half of the number that are in body cells, it's actually going to require two divisions of the cell. So the chromosomes are going to replicate once and then divide twice. Some major differences between mitosis and meiosis that I'm going to point out are how the chromosomes line up. During metaphase, instead of metaphase 1, instead of spindle fibers attaching to sister chromatids, they're going to separate the homologous chromosomes and pull them apart. During metaphase 2, the chromosomes are going to line up again, and then during anaphase 2, this time the sister chromatids are going to get separated. So, like I said before, there are two divisions, and first the homologous chromosomes are going to get separated, then the sister chromatids. The result is, at the end of meiosis, you're going to have 4 cells with the haploid number of chromosomes. This diagram is showing you the stages of meiosis 1 and meiosis 2, and one of the major differences that I want to point out, again, are how the chromosomes line up during metaphase 1 versus metaphase 2, and how the homologous pairs separate in anaphase 1 and the sister chromatids separate in anaphase 2. Another thing that I want to point out that I didn't earlier is that during prophase 1, you have small little bits of DNA that are going to do something called crossing over, meaning chromosomes are actually going to exchange little pieces of DNA with each other so that at the end of cytokinesis 2, what you're actually ending up with are 4 gametes, or 4 haploid cells that are very genetically different than the body cells. This is going to help create diversity amongst sexually reproducing organisms. This chart here is showing you a comparison, a side-by-side comparison of what would happen in mitosis versus meiosis, and how the daughter cells in mitosis are going to be genetically identical and have the diploid number of chromosomes, and in meiosis, how they're going to be genetically not identical and have the haploid number of chromosomes. Here's another side-by-side comparison of mitosis and meiosis. Again, you're always going to have one replication. However, in mitosis, you're only going to have one cell division. In meiosis, you're going to have two. Mitosis, you're going to end up with two daughter cells. Meiosis, 4. How the chromosomes line up in metaphase: sister chromatids are going to be on opposite sides of the metaphase plate and separate during anaphase. And remember, during meiosis, you're going to have two separations. Metaphase 1 is separating homologous chromosomes, and metaphase 2 is separating the sister chromatids. And the genetic relationship, by the end of mitosis, you're going to have two identical daughter cells, and meiosis, you're going to have 4 genetically different cells. Functions in the human body include repairing damage and growth from mitosis, and specifically making gametes from meiosis, making sex cells. Take just a few minutes and see if you can answer the following 3 questions on a half sheet of paper to turn them into your teacher at the beginning of the next class. What are the main differences in the purposes for mitosis and meiosis? Where does mitosis occur in the human body, and where does meiosis occur in the human body?